Good day, everyone. Thank you so much for joining us. I am pleased to welcome Dr. William Walsh back to the IBPF webinar series. Today, Dr. Walsh will give us an update on the latest research on nutrient therapy approaches aimed at normalizing brain chemistry in those with bipolar disorder. Also, we are very excited to announce that he will be sharing for the first time new theory findings. Dr. Walsh is president of the nonprofit Walsh Research Institute near Chicago and directs doctor training programs in the US, Australia, and Norway. During his 30-plus years as a research scientist and engineer, Dr. Walsh developed a science-based nutrient system that has helped thousands of patients challenged by bipolar disorder, in addition to many other conditions, and is used by doctors throughout the world. With warm welcome, we're presenting Dr. Walsh.
Well, good morning. Good morning, Debbie. Happy to be with you this morning.
Great, thank you.
Well, the title of the talk is Biochemistry Features of Bipolar Disorder and Advanced Nutrient Therapies. However, we are going to also reveal and explain to you what we think is actually going on, the underlying cause and mechanisms of bipolar. And of course, this is still theoretical. Organizations of public charity, a 501c3 public charity, located in beautiful Naperville, Illinois, where the sun is shining this morning. We are engaged in a number of activities: physician training, education, and research. The topics today, as Debbie mentioned, will include both the therapy that we've developed over the years for bipolar, and then our promising new bipolar therapy. So I've been doing this for about 35 years, working with patients with mental disorders, and we've had a lot of clinical experience. 10,000 cases of behavior in ADHD, more than 3,500 persons diagnosed with schizophrenia, more than 3,000 with depression, and about 1,600 diagnosed with bipolar disorder. So we have I think possibly the world's biggest chemistry database for bipolar disorder, that is involving blood and urine assays, and it's about 150,000 assays of blood and urine for bipolar patients. And it doesn't take a genius to look at this data and realize there are major chemistry differences between bipolar patients and the rest of society. So what do we find? It's very interesting that we find that some very high frequency imbalances that seem to dominate, and these are zinc deficiency. What we find that most bipolar patients are either low normal or actually deficient in zinc. Copper overload, folate disorder, which can be either too much or too little folate, Pyrrole disorder, methylation disorders, which can be either undermethylation or overmethylation. Essential fatty acids seem to be especially important in bipolar, and toxic metals sometimes are paramount importance in about 5 or 6% of cases. So what is the treatment itself? Well, it's a standard, not too unusual medical therapy. It involves a medical history and review of symptoms, and then special blood and urine tests to help identify what the chemistry is. And then based on the medical history and then a knowledge of the patient plus the lab work, we can then usually identify what chemical imbalances are present and make our diagnosis. And then we develop a nutrient program aimed at normalizing brain chemistry and normalizing neurotransmitter activity. Again, we don't give large doses of anything unless we need them to normalize blood levels and brain levels. So that's the basic system. Now, bipolar has been probably the condition that has caused me the greatest difficulty. We found it quite easy early on to find nutrient therapies that could help people with behavior disorders, with ADHD, depression and anxiety, and even schizophrenia and autism. We would do outcome studies where we would perhaps review 1,000 consecutive patients of the past, track them down and find out whether or not life was better, whether they were still doing the treatment, and to what degree they were still improved. And bipolar has always been one of the more difficult ones. It took us really, I think, 5 or 6 years before we even got to the point where 50% of them said that we'd really helped them. But where we are now, in the past 5 to 6 years, maybe the last even 10 years, the last outcome study we did showed that nearly 3/4 of the bipolar patients said after they had been doing this for a while that they were significantly improved. We've had hundreds who said that they were recovered and the symptoms were gone. But in most cases, we had I guess what you would call a nice partial improvement that they were quite happy with. However, we did strike out and continue to strike out about 1/4 of the time. I think the last number we got was 28%. We think we're probably a little better at this point. So about 3 out of 4 people who've done this after a year or so say that it's helped them, and about 1/4 say no, nothing good happened. So that's where we are. I think that measures up quite well to any outcome studies I've seen for pharmaceutical medications. Now, I get a lot of phone calls from doctors and who say, how could this possibly work? How can nutrients possibly help a mentally ill person, someone who might have a severe condition like bipolar disorder or schizophrenia? And often someone will say, well, don't you really need a powerful drug to get the job done? The answer is not really. Nutrients have power. I wrote a book a couple of years ago called Nutrient Power, and the reason I called it Nutrient Power was that was the, if anything, that was the one message I wanted to transmit to people, and that is that nutrients can have great power if you can individualize this, if you can identify that. And so the question is, where does the power come from? Well, if you look at the way where our neurotransmitters come from, the synthesis of our neurotransmitters in the brain and in the rest of the body, really the raw materials, most of the raw materials are vitamins, minerals, and amino acids. So they're directly involved in the synthesis of neurotransmitters like serotonin, dopamine, etc. And more recently, we've learned that they have a lot of power with respect to epigenetic gene regulation, the regulation of expression of genes, which is necessary in every part of our body all day, every day. And the dominant factor in depression and in anxiety and other mental illnesses very often is not the amount of neurotransmitter that's there, but the kinetics, the reuptake processes, the period of time that a neurotransmitter stays in the synapse before it goes back into the original brain cell. So this is where the power derives. And let's start with the synthesis itself, just to give a few examples. This chart shows the final step in the synthesis of serotonin, and it's enabled by a genetically expressed enzyme, L-amino acid decarboxylase, which is a mouthful to say. But the primary cofactor is vitamin B6 in the form of PLP, also known as P5P, pyridoxal 5-phosphate. And most people have all the B6 they need from their diet, and this goes rather well. But we find that in bipolar circles, in the population of bipolar people, at least 25 to 30% have a tendency for possibly very low levels of B6. So in this case, this can, if a person has a genetic or an acquired deficiency of B6, you can expect they're going to have low serotonin levels. Another example, and this one sometimes is even more important, this is the last step in the formation of norepinephrine, which is one of the major neurotransmitters. And all of your norepinephrine comes from dopamine. And it's a reaction that goes on in little tiny vesicles in your brain cells and in the rest of the body. And it's enabled by one of the more famous enzymes in biochemistry, dopamine beta hydroxylase. However, in addition to that, you need vitamin C, and the vitamin C is needed to maintain dopamine beta hydroxylase, keep it from disappearing. And but copper, divalent copper, copper plus plus, is the primary cofactor, and this reaction doesn't go well unless you have this copper, because the divalent copper actually binds loosely to the enzyme and enables it to work well. And normally people are supposed to have a good regulation of copper in your bloodstream. But people with bipolar disorder and other mental disorders, many of them don't have the ability to regulate copper. And if they happen to have an overload, for example, a really high level of copper, like many of our bipolar patients do, they tend to have excessive norepinephrine and not enough dopamine. And they've done, we've had research that's shown that you can get rather enormous differences in the amounts of norepinephrine and dopamine if that copper level is not what it should be. And if you have a copper overload, that tends to give you too much norepinephrine, and that is a recipe for a tendency for mania, for example. Another example is GABA. Now, there are many examples. I'm just going to show these three. This shows how GABA is formed. GABA is a calming neurotransmitter, and it's believed that many people with bipolar disorder have too little GABA activity. And again, in this case, yes, there's an enzyme, and it's also enabled by vitamin B6. Another aspect of GABA is that it's regulated. GABA neurotransmission is regulated quite a bit by zinc. So if a person is zinc deficient, they'll tend to be more nervous, have more neuronal firing, and more neuronal hyperactivity, and it's excitatory. And whereas they don't have the GABA to sort of quench that and calm the brain. So metal-metabolism disorders we find very frequently in bipolar. The most common of all is zinc depletion. We find most people that are bipolar do need zinc. Copper overload we find probably in a third of bipolar cases, and for these people, that it's absolutely essential that we normalize their copper level, and in that way we can elevate their dopamine and reduce their norepinephrine levels. And for some people, deficiencies of other metals are important, especially selenium, which is an antioxidant, calcium and magnesium, which are especially important in people who happen to have under-methylation imbalance. And then we also find some bipolar cases where toxic metals are dominant. It doesn't happen very often, but sometimes we've had bipolar patients who, one of the biggest issues was they might have had elevations of lead, mercury, or cadmium, for example. So let's talk about zinc. We recently did a study of 17,000 patients, and this one of my big databases, and I had a neuroscience intern study this, and with respect to the bipolar patients, those who had not been treated with nutrients, we found the median level was just 76 micrograms per deciliter. Whereas we know that the ideal zinc level in the blood, in plasma, is between 90 and 120. And our preference is for someone to be in the low 100s for best mental functioning. So we've learned that nearly all bipolar patients need zinc to function, to normalize their body chemistry and their brain chemistry. Pyrrole disorder is something that about 10 years ago was not too well known, but is now getting more and more attention throughout the world by researchers. And essentially, it is for most people, it's genetically transmitted. It's usually born in some people. It runs in families, and in effect, it is a rather massive double deficiency of B6 and zinc. And people that have this, among other things, have reduced serotonin, dopamine, GABA neurotransmitter levels, and they also have a tendency for extreme oxidative overload, too much oxidative stress, and depletion of glutathione, metallothionein, cysteine, SOD, catalase, which are some of these wonderful natural antioxidants that are supposed to protect us. So with oxidative stress, why do some people have oxidative overload? What can go wrong? Well, some of them have low natural levels of these protectors, glutathione, the metallothionein, and the others. I mean, these are chemicals that are wonderful in our body, and if a person gets exposed to mercury or lead, or something like that, then these protectors are supposed to bind to these and make them inactive so they don't harm us. But some people don't have they're not born with the proper levels because of genetic mutations. And a lot of people have this. Illnesses, injuries, emotional trauma can also cause oxidative stress. In fact, oxidative stress is necessary to help combat illnesses, for example. So it's not all a bad thing, but you can have too much of this good thing. And toxic metals, pesticides, industrial pollutants, those are examples of environmental exposures and that can cause oxidative stress to get too high. Now, in the brain, it's interesting that these are the three, I think, primary protections in the brain. Glutathione is the first line of defense. Metallothionein, which is a genetically expressed zinc-dependent protein. It's nature's backup system because we don't have a large supply of glutathione, and we get our glutathione from our diet. And so if a person has quite a bit of oxidative stress, you can run out of glutathione rather rapidly, and then metallothionein takes over, and they work hand in hand. They work together. It's nature's backup system. And then selenium is needed to make these this whole system work. It increases the kinetics of the glutathione and metallothionein antioxidant reactions. Now, in the last 5 or 10 years, we've learned a lot, and we've been able to improve all of our therapies considerably, and a lot of it has to do with recent advances in understanding methylation and epigenetics. So let's start with methylation. And I'm not going to go really deeply into it. I think a lot of you may already be quite familiar with it. Maybe there are others who are not too familiar, but I'll just summarize it. There are 2 types of methylation disorders. Most people who've studied methylation are very familiar with undermethylation. And however, in bipolar, we find that whereas in general, there are 3 undermethylated people for every overmethylated person, in bipolar it's closer to 50-50. And so a person can be either overmethylated or undermethylated, and that has a lot to do with your brain function. Now in bipolar, 70%, a little more than 2/3 of all bipolar patients have either overmethylation or undermethylation. And that is part of their problem with respect to brain functioning, and then most of them report improvement once we can normalize their methylation. So what causes it? What causes undermethylation? Well, the number 1 cause has to do with enzyme mutations that are called SNPs, single nucleotide polymorphisms, which is a mouthful to say. But basically, genetic mutations in the methylation cycle. Now, every single human being, you might say, is a mutant in that we all have tens of thousands of mutations. We do. And in methylation cycles, I think every single person in our audience here has probably at least 100 mutations in your methylation cycle. And what they do is they tend to weaken the ability to produce a chemical called SAMe, S-A-M-E, which is the primary methyl donor in biochemistry in the body. Another thing that can go wrong is that a biochemical called SAH, or S-adenylyl homocysteine, if that gets excessive for any reason, that tends to really shut down most of these, or diminish at least, the methylation reactions, and there's more than 80 really important methylation reactions in the body. And then protein deficiency itself, in some cases, since the methyl comes from protein, comes from the amino acid methionine, if your diet is really deficient, then that can be part of the problem, but usually it's SNPs in the cycle. And this is a picture of the cycle, and the cycle is fairly complex. This only shows part of it. And as I said, your methylation capability comes from your diet. It comes from your amino acids, your protein, especially, well, specifically methionine, which is at the top of the cycle in the middle here. And your methionine then reacts with magnesium and with ATP with the assistance of an enzyme. to form SAMe, SAM with little e. And SAMe is a fairly unstable molecule that gives up its methyl pretty well. And so it's everywhere in the body, it's everywhere in the brain, and it is the donor that gives the methyl needed for these, so many dozens and dozens of really important methylation reactions. And this is a rather complex cycle that's been really well studied lately, and all these little ovals, these green ovals, represent enzymes, genetically expressed enzymes. Well, some of these enzymes have been weakened over time genetically. They have what's known as SNPs. For example, on the left, the MTHFR is one that's been studied probably more than the others, primarily because this methylation is called the folate remethylation cycle, that little circle on the left, and that happens to be the rate limiting step in the whole methylation cycle. Well, MTHFR is a huge protein. It's got more than 500 amino acids, and a SNP basically means that one of those amino acids is in the wrong place. Instead of having one amino, it's got, so it's only like you might have a 499 of them in right, where they're supposed to be in one other place. And that's what a SNP is. And what these SNPs do is they have a possibility of weakening that enzyme. And the MTHFR SNPs that especially can do that, like the 677T and the 1298, are two of the SNPs that just happen to be in a location where it can affect the cycle. But anyway, the point of this whole cycle is that this system works quite well for most healthy people, and it provides a steady amount of SAMe, which can be that can carry out all these important methylation reactions. Well, how about overmethylation? How can we get overmethylation? And the answer is if we have impaired SAMe utilization. If all of the SAMe we're producing in the methylation cycle, if we're not able to use it like we're supposed to, we can have a glut of SAMe. Well, it's interesting looking at what happens to the SAMe. There, again, are more than 80 important methylation cycles, but it turns out that one of them dominates, one reaction, and that's the reaction creatine synthesis. And in many people, 70% of all of your methyl goes to that one reaction, and only 30% to the others. This can vary from person to person, and there are some people where only 40% of their SAMe goes to creatine, but in general, it's more than 50%. So let's just look at that a bit. So the synthesis of creatine is where most of our SAMe is used, and it starts with the reaction of two amino acids, arginine and glycine, which react with the assistance of an enzyme, AGAT, to form a chemical with a funny name called guanidino acetate. And then this guanidino acetate then reacts with SAMe. It's methylated to form creatine. And again, there's another enzyme, GAMT. Well, there can be SNPs or weaknesses in those enzymes. So you've got people have the possibility of weakening that can cause overmethylation or undermethylation, and I call this a methylation tug of war. So basically, what you have are for enzyme mutations, and believe me, they're prevalent. We now know that there are more than 10 million SNPs in the genome that have already been identified. And most human beings have tens of thousands of SNPs. But anyway, you've got SNPs that tend to, like MTHFR and others, that tend to cause undermethylation, and those that tend to pull in the other direction, that tend toward overmethylation. And it's really important for a patient, including a bipolar patient, to get the diagnosis right and find out whether or not they're overmethylated or undermethylated. And we've gotten really quite good at that. The genetic testing that some people are trying to use for diagnosis, so looking for SNPs, for example, and MTHFR and others, so far that's not been highly reliable, because we found people with MTHFR 677T homozygous SNPs who actually were overmethylated because they even though that tended toward undermethylation, it was overwhelmed by the other side. So, but fortunately, there are some lab tests that you can do, and a careful medical history. we can confidently diagnose whether a person is over-methylated or under-methylated. And that's an important factor. Again, more than 70% of bipolar patients have a methylation disorder, one or the other. So there's other features that we thing that's really striking is that virtually every person we see has high oxidative stress, oxidative overload. It's also clear evidence that nearly all of them have high levels of brain inflammation. So these are distinctive features of bipolar. So now let's talk a bit about epigenetics. 10 years ago, very few Americans ever heard of epigenetics and knew what it was. But now I think most of us are getting more and more familiar with it. And it's really causing a revolution in brain science and also, by the way, in nutrient therapy. Now, what is epigenetics? We all have our own distinctive, unique DNA, unless you've got an identical twin. And our DNA, we know, is responsible for the color of our eyes and maybe our ethnicity and whether we're short or small or tall and whatever. But what a lot of people don't realize is that your DNA is working for you every day, every second of every day, and it's nourishing every cell in the body. We've got about 37 trillion cells in the human body, and 25 trillion of them roughly have DNA. For example, your red blood cells do not. But so you have these trillions of cells throughout your body, and they are nourished by gene expression, by amino acids that are genetically expressed that nourish these cells from within. Now, your DNA contains about 20,000 genes, although the latest numbers are closer to 18,000. And every gene has just one job, and that's to make one specific protein. The problem is that every cell in your body, whether it's your liver, your heart, your eyeballs, wherever, your skin, you have the same identical DNA, but every part of your body needs different chemicals. And so every tissue, organ, and even every cell line needs a unique combination of proteins where a lot of these proteins are shut off because you have to have the right recipe for every part of the body. And this is remarkably accomplished by epigenetics, by gene regulation that occurs before you're born. There are two primary epigenetic processes, and the first one is what happens before you're born, primarily. And there are areas of your DNA which are called CPG islands, is what they call them, but they're areas near individual genes, and if they get highly methylated, it tends to shut off or downregulate the amount of that particular protein. And this is all set up as the body, as the little tiny fetus differentiates into arms, legs, and brain, liver, kidney, etc. And so it's all set up where you get hopefully the right mixture, the right ingredients for every part of the body. Now, the DNA in the nucleus of every cell in our body, they wrap gently around proteins that are called histones, and histones are composed of linear proteins, a ball of these proteins, 8 of them actually. And this is how the DNA, which is incredibly fragile, you can imagine, they're just individual molecules, and it helps stabilize them and forms a support for them. But we've learned that histones can actually be modified. It was thought for about 20 years that the histones were just there to support and be support for the neurons and the DNA itself. But you can actually change chemical reactions and gene expression by what reacts with the histones themselves. Anyway, and most of the epigenetic therapies that are going on now, there's a lot of interest in cancer research because we now know that most cancers are epigenetic. And the most of the adjustments that we can make in changing gene expression to help a person involves with histone modification. Most of our in utero methylation, item 1 here, most of us, these methylation, they call them bookmarks on our DNA. Once these are established before you're born, you tend to have those the rest of your life. Although they can change if you have environmental insults, for example. Okay, so let's I'll briefly go through this. I think some of you already know all of this, and a few of you may not. So I'd like to at least summarize it briefly for those who are not aware of this. Established in the womb, and this methylation at these CpG islands near the genes regulates protein production for each tissue. Environmental insults can disrupt this, and there are epigenetic disorders that can be formed. And one of these is cancer. I'm quite certain that autism is epigenetic. I'm quite certain that post-traumatic stress disorder is epigenetic, caused by environmental insults. And anyway, the DNA methylation bookmarks are usually permanent. So let's talk about the histones, because this is where the therapy aspect seems to reside, at least at present. These are support structures for the fragile DNA. They're composed of 8 linear proteins. They're twisted together like a ball of yarn. And I'll show a picture in the next slide of what that looks like. And originally believed just to serve only as structural support for packaging of the DNA. But now we know it is found that you can inhibit or promote gene expression depending on chemical reactions at what are known as histone tails, parts of these histone proteins that stick out of the ball. And this is a picture of a histone. And again, it's a ball of 8 linear proteins, and yes, you can see some of these tails sticking out of the core. And the DNA wraps around between 1 and 2 times around these histones. So, with respect to therapy, one thing we've learned is that the most important and the most dominant reactions at these histone tails are either methylation or reaction to form acetylation. So really it's a competition between methyl and acetyl groups at these histone tails. And if acetylation wins the war, if you have more acetylation in a particular area, that tends to promote gene expression. And if methylation happens to be what happens in a particular area, that tends to inhibit that particular gene's expression, or that amount of that particular protein. And we now know that nutrient therapy can modify these histones, that can react with the histones, and we can adjust the methyl-acetyl ratios and these reactions, and we can alter gene expression. And this gives us a capability we've never had before. We can now actually have a roadmap with nutrient therapy that tells us how we can manage gene expression of enzymes, and we can directly affect neurotransmitter reuptake with this knowledge. Now, just to get into it a bit, gene expression is laid bare. It has to uncoil from these histones. And the reason is that in your nucleus, where your DNA is, you've got molecules swimming around, large molecules called RNA polymerase and transcription factors that are big molecules. And however they're needed, they have to directly contact the DNA in order to make a protein. And these are constantly looking for exposed DNA to make a protein. Now, if you ask why does the DNA actually gently attach to these and wind around the histones, and the reason is electrostatic attraction. DNA is a weak acid. The histones are bases. And as we know, opposites attract. So you get this gentle attraction. Now, if you have an acetyl group that attaches to the right histone area, this will cause, it will lower the pH of the histone. And that causes the DNA to uncoil because you don't have as large an attraction. Whereas if methylation predominates for that gene, that increases the pH, and so you have a stronger attachment and DNA unavailability. and to display that, this is an example where acetylation won the war. And this is an area of your DNA, and what's happened is that because methylation is low and acetylation is high, this promotes gene expression, and as you can see, the exposed DNA and any genes that are in those exposed area are very likely to express genes. So methyl and acetyl ratios and nutrient therapy and even drug therapies can alter this and can regulate expression of some very important enzymes. This is what happens if methyl wins the war. Methylation tends to, in general, there's exceptions, but it tends to increase the attraction between the histones and the DNA, and it all jams together, and you get what's known as closed chromatin. Chromatin basically consists of two things: the histones, the proteins, and your DNA. And if this is all jammed together, then you will not have genes within that area expressing. Now, as we got into this, we learned that even though the acetylation occurs from the donor is acetyl coenzyme A, that's a chemical that's throughout the body, and SAMe, which is also everywhere in the body and every cell of your body. It's the amounts of those chemicals you would think might be really dominant factor, but really they're not. We've learned that it's the enzymes, the acetylases, deacetylases. An acetylase, for example, is an enzyme that enables that acetylation reaction to go on. And a deacetylase is a reaction that tends to remove the acetyl from that part of the DNA. Anyway, there are methylases and demethylases, and it's really these enzymes that dominate gene expression. And that is the focus for our nutrient therapies. We have to focus on that because that dominates far more than the amounts of SAMe or acetyl at the DNA areas. One of the most important areas in mental health, of course, is neurotransmission. And up until 1985, most neuroscientists thought that it was the key factors were the amount of serotonin and dopamine and the various neurotransmitters. But by 1985, the pharmaceutical companies and university researchers learned, no, the dominant factor is reuptake. The speed or the kinetics with which neurotransmitters, once they're sprayed into a synapse, the speed with which they return to that original cell. It's called reuptake. And so there's been a lot of studying of reuptake. And focusing on these things called reuptake transport proteins or transporters. And SERT is the serotonin transporter, and DAT is the name given to the dopamine transporters. And now these are proteins genetically expressed that reside in your presynaptic membrane. And these are proteins that once a cell fires can really quickly remove the neurotransmitters from the synapse. It can happen really fast. And it happens because of electrostatics. The neurotransmitters themselves have a charge. And so this can happen really quickly, or it can be slowed. This dominates neurotransmission of serotonin and dopamine, norepinephrine, and a few other neurotransmitters. But so the SERT and DAT expression, we know that depends primarily on the acetyl-methyl competition that we talked about. And nutrient therapy can alter serotonin and dopamine reuptake. I just want to mention SSRI antidepressants. The way they work is they go quickly into the brain, and they interact with these transport proteins, and that's how they work. They tend to partially disable them and slow the return of the serotonin and the synapse back to the cell, so the serotonin stays in the synapse longer, and you get more neurotransmission. So we've already learned a lot. It seems like every month there's so much research now on epigenetics because it's so important. This is what we already know enough to help a lot of patients, including a lot of bipolar patients. But here's some of the things that we already know. We know that vitamin B3, also known as niacin or niacinamide, we know that reduces dopamine activity. We know exactly why. It tends to disable a an enzyme that's called a deacetylase. So it acts as what's known as a deacetylase inhibitor. And what this does is it tends to knock dopamine down. This is really important for a patient who happens to have excessive dopamine activity. We know that methionine and SAMe, which are nutrients over the counter, they act as serotonin reuptake inhibitors, just like SSRI, only in a different mechanism. And so therefore they can increase serotonin activity. So if a patient has that problem, if low serotonin neurotransmission is an issue, these nutrients can be very helpful. Folates can be either you can either be a deficiency or overload, and we now know that folates, because of epigenetics, they tend to sharply reduce serotonin and dopamine and norepinephrine neurotransmission. They act as serotonin reuptake promoters. So we have to be careful not to give folates to a patient who has a mental disorder that really involves low serotonin activity. Because even though you might benefit methylation with a folate, you don't want, the last thing for those patients that you want to do is knock their serotonin activity down. We know that biotin can repress expression of certain genes. Zinc interacts with DNA and increases metallothionein expression. Interestingly, resveratrol, which is in the news a lot, and curcumin, it's now clear, there are peer-reviewed journal articles that show that it suppresses histone acetylation. And many people think that that's why resveratrol and curcumin can be really helpful to a patient. Genistein, another lesson in the news, it enhances expression of certain genes. And this is a partial list. This is growing almost every week and every month. And we're learning a lot about how nutrients can really impact brain function. And this is all quite new, and it's very exciting. Now, the therapies that we've been using so far are really strictly, so far, have been focused on histone modification, causing to either cause the DNA to uncoil or to get compressed. But there are advanced therapies that have been discovered in cancer research, where most of the epigenetic research is going on. And there are therapies now where you can actually alter these epigenetic marks in histones. There's just early basic research on what they call DNA editing, where you could actually snip out a piece of DNA that may not be helping a person and replace it with a proper DNA, part of that snippet of the DNA. A lot of effort now is going into bromodomain therapies for cancer, and I think there's one that's already in phase 3 FDA trials. And bromodomains and chromodomains, these are parts of these transport proteins that we talked about. And what they do is for a gene to make a protein, you not only have to have the DNA exposed and interacting, but you have to be able to identify and it's really these bromodomains and the chromodomains that can identify, they can read the molecules that may have attached to the DNA or to the histone. And then the microRNA therapies are also becoming really important, because microRNA, these are tiny, short snippets of RNA that once a protein has been made and is ready to leave the nucleus and to go down into the cell reticulum and make a protein, I call microRNA the traffic cop. It can just stop it cold and stop it and prevent it from leaving the nucleus. So these advanced therapies, I think, are the future, both for cancer, post-traumatic stress, and I think very likely also for bipolar. There's high complexity and technical difficulty, but there's active research, and we're learning so much. I think this is really high promise for the future. So just a summary, just to summarize the nutrient therapy aspect of my talk, there's been significant improvement. And presently, our last outcome study showed 72% patients said that they had significant improvement And we think that that's going to get a lot higher as we get smarter and just learn more, especially with epigenetics and methylation. The sciences of methylation and epigenetics have already led to improved clinical outcomes. And I think epigenetic therapies of the future offer really great promise. Well, now we'd like to switch to another topic. I've been working with Dr. Robert DeVito, who is a noted psychiatrist, and we've been working for a couple of years now trying to understand better what bipolar disorder is and maybe develop an understanding of what the mechanisms and the causes of it are. Dr. DeVito was at one time served as the chairman of the psychiatry department at the largest medical school in the Chicago area, and he also served as the director of psychiatry for an enormous hospital at Loyola Medical Center. And he's been doing research for years, and the two of us are good friends, and we meet about once a week. And this is one of our topics is, what is bipolar, and can we understand it better, and therefore maybe help people? Well, what are the distinctive features? Well, there's a lot of variety and various types of bipolar, and different chemical imbalances that might be prominent in one biotype and others. But the one thing that the distinctive feature, first of all, episodes of mania, which is you might think of as hyperactivity of brain neurons in many areas of the brain. And then, of course, episodes of depression. And for bipolar 1, persistent cycling between mania and depression. And we've been focusing mainly on bipolar 1. So what are the really important questions? What are the mysteries that need to be solved? Number one, what are the causes and what are the mechanisms of mania? What on earth is going on in the brain that causes so many brain cells to be firing, to be excitable and excessively fire? What causes that? And what's the mechanism of it? And if you were to do a brain scan of somebody in a manic phase, it would light up like a Christmas tree. You'd have an unusually high amount of brain cells firing. The question is, why? And then, why is it that prolonged manic episodes transition into depression? What's that about? Why? And then, what causes mania to reappear after an extended period of depression? These really have been, you might say, mysteries, and you really need to understand all of them. So we believe that a lot of this, most of it for bipolar, has to do with neuronal hyperactivity, with brain neuron voltages. Mania is associated with greatly increased neurotransmission throughout many areas of the brain. We know that. What causes a neuron to fire? Why does any neuron fire? Well, neurons are like little batteries. Our brain neurons are like little batteries, and it's got a lot of potassium outside the cell, sodium outside the cell and potassium within the cell, and this creates a voltage. And it's a little less than a tenth of a volt. And what happens is that if that voltage diminishes to the point where it reaches a threshold, then it's like a gun going off. It fires. The neurons don't have different degrees of firing. It's an all or nothing thing. If it fires, it fires like a bullet leaving a gun. And then the cell depolarizes, which means that the resting voltage of that neuron is lost. You lose that voltage, and it sort of reverses. But then they have the ability to very quickly reestablish that voltage. Now, of course, when a neuron fires, what happens also is that calcium ions rush into the cell and cause the neurotransmitters to be sprayed into the synapse. Well, neuronal hyperactivity can result if that resting voltage is diminished, if it tends to get closer to the threshold for firing. So let's just say a little bit about neuron voltage. Brain cell voltages form ion gradients across the neuron's membrane, with most of the potassium ions inside. By the way, it's about a 20 to 1. You have about 20 times more calcium ions inside the cell than outside, and most of the sodium ions are outside. And this is established by what they call the potassium-sodium pumps, and the power from that comes from the ATP, the energy from your mitochondria, and that's what causes these ions to be displaced. Now, so you've got potassium ions inside and sodium preponderance on the outside, and as we know, positives tend to repel each other, and that sets up the voltage. When a neuron fires, what happens is sodium ions rush into the cell, and potassium ions leave the cell, and it depolarizes. You lose that voltage; it's gone for a very short time. However, these brain cells usually return to the original state really quickly. It's called repolarization, and the sodium ions are quickly pumped out of the neuron. But the potassium ions it's very important that they get removed, many of them do not go back into the cell, but they have to be removed by astrocytes, by glial cells that surround the synapse and have the ability to just suck up all these potassium ions and back into the capillaries of your brain. And they're called M currents, and this is going on constantly in the brain. So what happens if you have neuronal hypoactivity? If your neurons tend to be firing lower than the usual rate throughout most of your brain, well, if you have reduced neurotransmission of serotonin, dopamine, norepinephrine, and NMDA, and other receptors, this can result and often results in severe clinical depression. So we believe that we've come up with a bipolar disorder theory that we're quite excited about, and we'll explain why. We believe that bipolar disorder may be caused by impaired removal of these potassium ions by these astrocyte glial cells after a cell fires, after the depolarization, and that since you don't remove all of it, you don't have as high a voltage once you repolarize, and that causes excitability and can cause mania. Well, this single factor, what really has us interested is that it can explain the development of mania, and the single factor can also explain why there's a slow descent into depression and then chronic cycling between mania and depression. This one factor, this one process. Now, it may be impaired by a multitude of genetic or environmental factors, but we think this may very well be the essence of what bipolar is. And it has to do with these things called glial cells. So I want to say a little bit about that. Glial cells are 85% of all the cells in your brain are glial cells. 15% are neurons. And the interest in this was very small until something happened with Einstein and Einstein's autopsy in his brain. And let me just briefly mention that. Einstein died in 1955, and at his autopsy, someone stole his brain. Four large pieces of his brain were missing and were not in his casket when he was buried. 30 years later, they turned up. They found out who had the brain, and they were preserved in formaldehyde. And people, of course, scientists wanted to study Einstein's brain to try to understand why he was so brilliant. And so they did. And at California, Berkeley, and other places, scientists started studying what was left of his brain. At first, they were disappointed because they found that there's not much difference in his neurons. He didn't have more brain cells than others, except in one small area. He had part of his, on the sides of his brain, they found that he had slightly more neurons in an area that has to do with mathematics, which makes some sense. But then they found that millions of others had the same kind of higher levels. However, what they found after they started delving into it, there was a huge difference in Einstein's brain. And that was that he had a much higher number of glial cells, especially glial cells called astrocytes. And because of that, people knew about the glial cells for 180 years, since the middle 1800s. But nobody ever thought they were important. They thought they were just structural support for the neurons. But by studying this, they've now found, by doing studies after death, they find there is a strong correlation between glial neuron ratios and IQ. It's definite. If you study people from Mensa, you would find that they would have a lot more glial cells than other people. And so there's been an enormous amount of intensive research just in the last 20 years since we've known this. And it really was Einstein's brain that really stimulated interest in this, and it has the possibility of actually revolutionizing neuroscience. The major types of glial cells are astrocytes. These are the larger ones, and they tend to directly connect brain capillaries and the neurons themselves. And all day, every day, they deliver nutrients to the neurons. But they also help regulate neurotransmission, because it's the astrocytes that pull the potassium away after a cell fires. It enables the cells to repolarize and get their voltage back. Oligodendrocytes, which was a really funny name, they have one job, and they form the myelin sheath. They're able to wrap their membrane in layers along axons and form the very important myelin sheath. And then the microglia are smaller ones, and people consider them the brain's trash collector, but they also have a role in immunity. But for our theory, it's the astrocytes that we focus on. Astrocytes and potassium ion removal. These astrocytes glia surround the synapses, and they quickly take up these potassium ions through ion channels. And so after neurons fire, it's really these astrocytes that enable the repolarization to reestablish the voltages. And they transport them to blood capillaries. The glia connect to each other by what's known as gap junctions. It's quite amazing how rapidly and how efficiently ions can go from one glial cell to another. Anyway, they just deposit the potassium into the bloodstream. These are called M currents. Now, impaired potassium ion removal can explain virtually all aspects of bipolar. So let's get into that. This is just a picture. It's not to scale, of course, but it shows how the astrocyte cell, which is in the middle, connects to both capillaries and neurons. And there's a constant flow of nutrients in and potassium ions out. And that's the avenue or the street by which it occurs. So we have a model of bipolar 1, and I'm not going to go into great depth. We believe that the onset can be triggered by a number of things. Oxidative stress. There are probably people born with a tendency for bipolar. We know that there's heritability in bipolar, that it does to some extent run in families. So people might have weakened potassium removal capability when they're born, and so they're more vulnerable to insults. There might be oxidative stress or some other mechanism that can weaken it to the point that you start to get incomplete potassium ion removal. So you don't get all of it out of the synapse that you need after a cell fires. Well, we know that if you don't have all of your or most of your potassium out of that synapse space, what you wind up with is somewhat reduced neuron voltage, and that tends to produce excitability and hyperactivity of a neuron, also known as mania. That can cause mania. So then what happens? Now you've got mania going on at that neuron, and so it's starting to fire more often than it should. It's hyperactive, and you're pouring more and more potassium ions into that synapse. Well, if you get enough potassium into that synapse, you get to the point where neurotransmission rates collapse, because you can't even establish a proper voltage for this whole synaptic activity to occur. And we know that if you have reduced neurotransmission rates, this is manifested as depression. So then if you've got this neuron now firing very slowly, if at all, now you're really able to start clearing up the potassium ions out of the synapse. And so guess what happens? You get to the point where you establish a voltage again, although before you get all the way there, you're now at the point where mania occurs again. And so that results in a return of mania, and this cycling can continue indefinitely, up and down, up and down. And well, I guess what has us really excited is that we now have a single mechanism that explains all aspects of bipolar 1 disorder. This is not just a theory. There's a lot of nice, really high-quality peer-reviewed journal articles. For example, genomic research. There's been genome-wide association studies that involve more than 35,000 bipolar patients and more than 20,000 controls, and they're identifying the genes that are not functioning properly in bipolar, and it's a fairly short list at this point, but they're finding that that definite implication of potassium ion channel abnormalities in bipolar. I mean, they see it clearly, and I think the p-value in one of the papers I saw was 10 to the minus 8. So it is at least one of the ways in which bipolar can be enhanced. So that's the end of my talk. I believe that even though I ran over by maybe 5 minutes, I think we may have time for questions, and so I'll leave that to Debbie, who will be receiving any questions you may have.
Excellent. Thank you, Dr. Walsh, for sharing your insight and information. We do have a number of questions, and some of the questions did come through early on, so just excuse if you have answered this already, but I'll go ahead and start with the first. I understand vitamin K2 prevents formation of free radicals in the brain. What do you know about this?
What prevents formation of free radicals?
Vitamin K2.
There are a lot of nutrients that can enhance protection against and can cope with it. We have really an army of wonderful natural chemicals that protect us from oxidative stress. And probably the number one that's well known and respected is glutathione, but there are at least a dozen really chemicals that do this, and they help each other. And if you have a weakness in one, the others tend to take over. Now, vitamin K2 is one where there's some evidence that it's one of the army, one of the good guys that helps protect us.
Great, thank you. The next question, you mentioned bipolar and autism are related. What are the similarities?
I didn't really say that they were related, but I think they may both be epigenetic in origin. Epigenetic disorders are disorders that are characterized by a person who might have relative normalcy, and then in a very short time, there's sort of like a breakdown or an onset of a disorder. And the disorder is a complex disorder because it has to do with a multitude of genes that are now not functioning properly, and the condition doesn't go away. Once you have the onset of the condition, like regressive autism, or those who have a bipolar breakdown, if you want to use that term, those conditions do not usually go away, and they can cause problems the rest of your life. That's really the only connection. It's not biochemical.
Great, thank you. The next question. I live in Denmark, and she's asking if you can get over-the-counter methylation tests done via postal service.
Well, I've spent a few years going to Scandinavia, and I've been there 3 times and trained a few dozen doctors who are able to do the therapies I mentioned. And I know we had at least a couple of doctors from Denmark, and these doctors are able to get the lab testing done. It's often done through through a person in Norway, Oslo. It's a lab by Dr. Dag Tveiten, Tveiten. So if they're interested in this, they could contact that laboratory and they would be able to arrange the testing.
Great, thank you for that information. The next question, related to gene regulation, has the vitamin D serum status been tracked in these interventions? What about vitamin K2 and MK7?
There's a lot of research on this, and it's just begun. We all know the importance of vitamin D and vitamin K2 and other, and most of the focus has been on other nutrients, and the reason they're in the news now is that people woke up and said, well, why don't we try these? And this is going to be a continuing process. Every single nutrient, I think, will eventually be studied with respect to whether or not it has a major impact on brain function. The focus mainly has been on neurotransmission, but then oxidative stress is so important to the brain, and inflammation also, that eventually this will all be known. And I think the reason they've been in the news lately is that it's only been recently discovered that they're important in this area. There's evidence that they too are related to these factors.
Great, thank you. The next question, are you stating that the nutrients merely synthesizing more serotonin and norepinephrine versus inhibiting reuptake?
No, they're both important. Of course, we have to have the neurotransmitters. In the case of serotonin, yes, you have to have a certain amount of serotonin. But the amount of serotonin itself doesn't vary dramatically from person to person, even people who have disorders. But the reuptake dramatically affects it. So really, you need to have enough serotonin. And if you take tryptophan or take other issues or take therapies, diets, or whatever that'll increase the serotonin levels, you can help a person somewhat. But it's a minor effect compared to the dramatic, powerful effect of reuptake. So it's reuptake dominates neurotransmission. So if you're doing a nutrient therapy or a drug therapy, the focus will usually be on reuptake and not on the amount of the neurotransmitter. We've learned that 30 years ago, actually. It was around 1985, and until 1985, drug companies and nutrient therapists were all focusing on ways to increase or diminish the amounts of these neurotransmitters. And starting in 1985, that's when they developed the first antidepressants that were SSRIs, serotonin reuptake inhibitors. And that's because they realized this is the name of the game. This is the way to really help a person or to really impact neurotransmission.
Great, thank you. What behaviors might be seen if dopamine activity is increased?
People who have high dopamine activity, back in the early 1950s, Abram Hoffer and others, in studying schizophrenia, found that the classic paranoid schizophrenics tend to have high levels of dopamine, and their therapies, which included, for example, niacin, we now know why they work. These are dopamine. They tend to lower dopamine activity. They're dopamine reuptake promoters, and so if you have high dopamine, we know that low dopamine is associated with ADHD, and that's why people give amphetamines to drugs to try to help that, because they elevate dopamine activity. But if you've got too much dopamine activity, that can be associated with anxiety, sleep problems, and it's associated with many forms with one particular form of bipolar. We find there are, and we call this the over-methylated biotype of bipolar, and these particular people have too much serotonin, dopamine, and norepinephrine, and you can really assist these people if you can treat them effectively and get those normalize those neurotransmitters, the neurotransmitter activity.
Great, thank you. The next question, do you have suggestions for clients who cannot tolerate B6 and P5P due to enhanced anxiety? Is there an SMP that leads to lower levels of these vitamins for clients who are on a good diet?
We have run into that a few hundred times. We will test someone and do a medical history, and then we will clearly see. We definitely know that these people who have, usually they have extreme anxiety, and we find very often that they report intolerance to even very low levels of B6. However, you have to recognize we've all got lots of B6 in our body. So how can you be allergic or intolerant to something that's already in your body? What we do in these cases, I think it's the anxiety itself that can cause this. I mean, people with extraordinary anxiety, they can get all wrapped into a higher anxiety state whenever they take a therapy that they think might help them. And I've done research studies on them and given these patients placebos that had nothing in them, and I remember one I did with a high anxiety patient who said that she was intolerant to B6, and so we alternatively gave her placebo with nothing, and then gave her B6, and she said the worst side effects she had were with the placebo. So there's that aspect, and it seems to be specifically with anxiety. So what we've learned is we can actually get these people to tolerate the B6 that they really need. And we have to start with tiny dosages, maybe as low as 2 or 3 or 4 milligrams a day, every other day, and then just slowly, gradually build up to tolerance. And it almost always is successful. And life is better once these B6 deficient people get their B6 normalized. But yes, there are some people who have extraordinary difficulty getting to first base, and you don't want to give them the full dose of B6 to start with, or they will report it's the worst day of their life. So we can cope with that. If they're not truly intolerant, if they're B6 deficient, they need it, and it's just going to continue. Now, the average person gets quite a bit of B6 from their diet. This happens every day. So no, they're not intolerant to it. They just need to raise the level very slowly.
Great, thank you. Next question. I have pathology tests that show both levels of SAMe and SAH to be raised. How could that be? Also, some patients show COMT double positive and MAOA double positive.
Well, these mutations that were described can happen. They happen to a lot of people. As I said, there are more than 10 million SNPs in the human genome, which means our entire DNA strand. And we all have a different SNPs, different mutations. Let's see, can you repeat the first part of the question? Oh, the person was high, had SNPs. They had testing that showed they were elevated in SAMe and in SAH. Well, that happens a lot. If your SAH is elevated, it tends to dominate everything. And in that case, it's the ratio for those people. If you've got both an elevation of SAMe and an elevation of SAH, it's the ratio of SAMe to SAH that dominates. So whatever that ratio is, if this is a doctor's data lab result, you can look at the bottom and you can look and see what that ratio is. And if that ratio is low, that means undermethylation. And if the ratio is high, it might mean overmethylation. Now, fortunately, we know the symptoms and traits that you find in most people that are overmethylated or those that are undermethylated. So the diagnosis is not that difficult. And I really like this test. But that's what happens if both are elevated. It's kind of unusual if they're both elevated. More often you see them both depressed, or one high and the other not. Now, with respect to the COMT, the other SNPs that were mentioned, again, these SNPs may or may not weaken enzyme or protein production. They may or may not, or the functioning. It's really the functioning of the enzyme. The enzyme might be produced by gene expression properly, but because it's altered, because it has a SNP, because it's got one or more amino acids in the wrong place, they may have weakened function. If you've got a comped homozygous comp SNP, what that means is that you're likely to have significant weakening of that particular reaction, of whatever reactions involve comp. That's all it means. It may or may not be significant. We've seen dozens and dozens of patients who had what appeared to be important SNPs that were homozygous, like the 677T.
Great, thank you. The next question.
And in some cases, it doesn't seem to affect them. One way to see whether or not methylation SNPs have really impacted you is to take a look at your homocysteine and your SAH levels. If those levels are quite normal, we've seen people with a 677T homozygous SNP who had normal or even somewhat depressed homocysteine levels. Well, for those people, it's not nearly as important as if they happen to be high. Any other questions? Can you still hear me?
Sorry, I think we had a little difficulty with our sound. Can you hear me now?
Oh, I can hear you now, yes.
Okay. Has anyone looked into the vitamin D levels in the research you've done, since it regulates 17% of the DNA?
Well, it's involved in regulation of the DNA. It's one of the factors, and it's an important factor. And thank God, mainstream medicine has discovered vitamin D, and it's now being tested by more and more doctors. There's some controversy about what is the correct amount. When I go to Scandinavia, it's amazing how low the vitamin D levels are. You can imagine where they have so little sunlight. They tend to be extraordinarily needy as opposed to people closer to the equator. But we believe that the ideal level is somewhere between 30 and 50 in the usual units. There are some people who believe that you're better off if you have like 80 or 100 level, and I think that's a little bit risky. Especially if a person has any problem with liver function or elevated liver enzymes. But the target usually is around 50. And now I think it's a rather easy thing for doctors to manage. And I think this is something that is really great that mainstream medicine has discovered this. I hope the next nutrient they discover is zinc, and then B6 and on, because normalizing each of these is so important in brain function.
Great, thank you. The next question, how much zinc and vitamin B6 would you recommend taking as a supplement if a patient is already on taking lithium?
Okay, I think the amount of zinc one takes is the amount it takes to normalize their plasma zinc level. We know what normal and healthy is, and what we'll normally do, the doctors that do this would make the best estimate they can of the dose of zinc that they think is likely to normalize their zinc. Maybe try to get it in the 100 to 120 micrograms per deciliter zone. But then after 3 or 4 months, then they would retest it and see what the level is, and then adjust the doses up or down, because then they can fine-tune the dose to make the level normal. Again, we're normalizing the level. And the same thing can be done with B6. And so the levels, the proper dose is the amount it takes to make a person no longer have an imbalance in that nutrient, to normalize it. And it's different for every person. And some people don't need any. And if a person has manic episodes, one would try, I think, to make sense to try to do whatever nutrition-wise or even diet-wise that can tend to combat the mania, to tend to increase neuron voltages, things like GABA, for example. If that interacts with brain cells, it tends to increase the voltage potential and therefore sort of relax the cells, make them less excited. Glutamate is the opposite. Um, so I think that in terms of nutrition, anyone who has bipolar disorder, I think, this is not something you should do on your own. You shouldn't do this at home. You really need to have a doctor experienced in nutrient therapy manage and supervise this. I urge people not to do this by themselves.
Great, thank you. The next question is, do you have any information regarding the link between glutamate and depression?
Well, of course, glutamate is the #1 neurotransmitter in the brain. If you look at all the neurotransmitter or neurotransmission, there's more glutamate receptors and more glutamate neurotransmission than any other. Now, glutamate in most areas of the brain is excitatory. So glutamate tends to cause neuronal hyperactivity. And there's one exception, and that is at what's known as the NMDA receptor. And in that case, low levels of glutamate are the usual problem for people who have that issue, and that includes quite a few people with bipolar disorder. So for them, you don't want to give them glutamate because that would make the rest of the brain tend to go a bit crazy. So what you do in that case is the NMDA receptor, fortunately, there are nutrients that, especially D-serine and D-cycloserine and other, and glycine, there are nutrients that can help adjust and normalize the function of that important neurotransmitter. But glutamate itself tends to cause mania, and that's something especially to try to minimize if you're in the midst of a manic episode.
Great, thank you. The next question, by regulating potassium and providing nutrient therapy to normalize brain chemistry, can oxidative stress be curtailed, thus improving long-term prognosis?
Well, I think you absolutely have to deal with oxidative stress with every bipolar patient. I've personally been involved working with my medical colleagues with 1,600 cases of bipolar, and we find that we must deal with oxidative stress and do whatever we have to either normalize it or make it far less significant, because we're certain that is part of the problem. And regardless of the reason, it's an outcome of bipolar. In most cases, it's a distinctive feature, and so there are many ways one can do that. For example, if a person has a zinc deficiency or insufficient zinc, that tends to cause oxidative stress. A Pyrrole disorder, wow, that's a way to really have oxidative overload, is if you happen to be born with a Pyrrole disorder, and we know how to correct that. So it's really depends patient by patient specific, but there are wonderful antioxidants, including things like vitamin C and vitamin E and selenium and NAC and on, that are wonderful at combating it. But if a person has bipolar disorder, you can be sure that they have both inflammation and they have excess oxidative stress, and I think that you need to address it.
Excellent, thank you. I think we're going to take one more question, and that would be, how does heavy metal detox play into therapy?
Well, we've, again, studied more than 30,000 patients with a mental problem, including more than 1,500 with bipolar, and we have occasionally found people who have, appears that for them, toxic metals, like usually it's mercury or lead, sometimes cadmium, that seems to be a dominant factor for them, but it's only about 1 out of every 20 cases we've seen. However, we do find that you generally tend to have not necessarily heavy metal poisoning, but heavy metal overload in probably at least a third of bipolar patients. So it's really important to normalize that. Now, most of the toxic metals leave the body rather rapidly. The half-life of mercury in the body is 42 days. The half-life of methyl-ethyl mercury in the brain is around 70 days. So even if you did nothing, it would eventually leave. assuming you don't have additional exposure. Lead, on the other hand, the half-life of lead in the body is about 22 years. So if a person has lead overload or lead poisoning, they're going to have to deal with that the rest of their lives because so much of the lead will be resident in their bones and will continue to cause problems.
Great, thank you. I know that we have many more questions that are unanswered. I'd be happy to receive them via email and then share them with Dr. Walsh if he's available to answer. My email is dbrown@ibpf.org. And lastly, I want to thank you, Dr. Walsh, for this incredible information. There's a lot of interest, and you see his contact information on the screen. And I will be happy to share any resources in terms of how to find someone to connect with in your neighborhood in terms of the nutrient therapy programs, research, doctors, etc. Thank you again, and here is Dr. Walsh's book up on the screen to remind you, and I hope to see you at the next webinar, and have a great day, everyone.
Thank you, Debbie.
Thank you.